Hi, my name is Sydney Eiland. I'm a junior at the college. This summer, the SURF grant afforded me the opportunity to work under Dr. McElroy in his biology lab. I spent the last 2.5 months creating 3D models of lizard pelvises. This project is called Lizard Hips Don't Lie, Pelvis Skeletal Anatomy in Lizards. This image that you see right here is actually one of my finished products of one of the 3D models that I did create. The ultimate goal of the project is to have the 3D models of both the bony pelvis and the surrounding musculature in order to observe the variation. Then we'll explore how that variation evolves and explore the functional and ecological correlations of variation in shape. We know that form impacts function, and this gives us a very detailed look at the form, the anatomy, and eventually we will digitally dissect the specimens. I used a program called Slicer 3D to create 3D models from CAT scans. There are features that allow you to paint pixel by pixel and build the structure. This right here is a screenshot of a portion of what I see when I'm on the program. It shows you the axial, sagittal, and coronal views of the lizard. So using these 3 planes, I could figure out with a lot of trial and error which structures we wanted to be part of the 3D model of the pelvis. The following slides are views of the 3D pelvises that I created. This slide right here shows the anterior view of the 4 species that I was able to finish this summer. Just from these initial images, you can already see that all the pelvises are very different. I included an image of the lizard itself underneath each pelvis for reference of what the species actually looks like. This slide shows the lateral or the side view of each pelvis. And here is the posterior views of each pelvis. Again, you can see how different each one looks, even in the 2D views. There are quite a few anatomical differences that I could report in great detail on. However, for the purpose of this presentation, I've narrowed it down to 3. The ilium length and shape is important to note because it is directly related to the lizard's mode of locomotion. The ilium is the principal insertion for knee extensors, which are key in powerful movement. The longer or broader the ilium is, the more space for the knee extensors. You can see with these 2D images how different each ilium looks. And I'll go ahead and circle what the ilium actually is if you're unfamiliar with the anatomy of pelvises. And just looking at that, again, you can see how different they are in width, in shape. The next structure I wanted to point out is the preacetabular process. If you look at the first 3 species, they all have some kind of bony process inside the red circle on the image. However, if you look at this last species, the Ryopa, it does not appear to have one. This process is where the quadratus lumborum originates to connect the appendicular and axial skeletons. It's a muscle that's responsible for elevating the anterior or the front part of the body, and it's very useful in bipedal and branch-to-branch locomotion. Also, the iliopubic ligament attaches to the preacetabular process, and it connects it to the pubic tubercles, which is a structure on the anterior pelvis. It also encapsulates different nerves and vessels, as well as it provides an attachment for the puboischiotibialis. Finally, the cartilages appear to be very different. Looking at these images, the cartilage is anything colored red. There isn't much literature available on the functions of the different cartilages, but we do hope to investigate them in our lab now that we have the anatomy down. All 4 species have a present epipubis here at the top, and you can see that the Ryopa actually lacks proischiatic cartilage, so you can see the Callosaurus, Agama, and Phrynosoma have this cartilage right here, but the Ryopa is lacking it. Also, all the species have the hypoischium, and we do know a little bit about that, and that's that the hypoischium is thought to connect to the tail. Continuing the project, we have several more species that need to have 3D models created using the same methods. We'll soon have iodine-stained CAT scans that will enable us to see all of the connective tissues. The musculature and connective tissues will be overlaid with the bony pelvis and then digitally dissected. Eventually, we'll be able to quantify the variation amongst the pelvises and actually be able to define their different morphospaces.